Hello, I'm Cassandre Mahe, and the study I'm presenting today is part of my PhD project, supervised by Féron and Gheusi from the Université Sorbonne Paris Nord in France. It focuses on the effects of family structure on isolation calls produced by pups in a monogamous rodent, the mound-building mouse. In this species, females and males usually form long-term bonds, each partner contributing to the care of the young. Cases, however, exist in the wild where the male comes to disappear, leading the offspring to be reared by the mother alone. In addition, some related females remain together after weaning. A male may then join them and reproduce with each of them, leading to a polygynous structure where parental care may differ from the previous ones. It has been shown that such deviations from the common family structure have consequences on the reproductive success of the parents. But little is known about the effects it may have on the behavioral profile of the offspring. This is what we investigated in this study. One of the earliest behaviors that can be tested to assess the behavioral profile of the young is isolation calls. These calls are ultrasonic distress vocalizations produced by pups in an anxiogenic situation, typically when isolated from the parents and siblings. Contact with a familiar adult before isolation can strengthen or potentialize this call response, a phenomenon known as parental potentiation. The pups tested in this study were raised in either one of three family structures: in monoparental units with the mother only, in biparental units, including both parents, or in polygynous units with the two parents and an additional adult female. On postnatal day 9, isolation calls were recorded following this procedure. All pups in the litter were tested in a random order. A pup was isolated from its home cage and placed in the apparatus for 5 minutes, during which its vocalizations were recorded. Right after this first recording session, the pup was returned to its home cage, which usually leads the adult to interact with it. And then, in order to assess the effect of parental potentiation, it was once again isolated for 5 minutes. The results of this procedure are presented here. You can see the mean number of vocalizations produced every 10 seconds of the isolation phases by pups from the monoparental units in blue, the biparental units in yellow, or the polygynous units in gray. Two main results stand out from this graph. First, as you can see, pups from the monoparental units call significantly less than pups from the polygynous units. Second, we did find a rebound in the mean call rates between the end of the first isolation and the start of the second isolation phase. This rebound matches the parental potentiation phenomenon, which is not affected by the kind of family structure the pups come from. Parental potentiation has not been tested in this species before, so it is an interesting finding. How to explain these differences? We suggest several interpretations of these lower call rates in the pups from monoparental units. First, it is possible that these pups, being more often alone in the nest, are more habituated to isolation, making it a less anxiogenic situation for them. Alternatively, this could reflect a difference between the coping style of the pups when exposed to an environmental challenge, with pups from monoparental units being less proactive than the others. Further analyses are needed to determine how to interpret this data. Analysis of the parenting behaviors during the first postnatal week in each of these reproductive units and the assessment of behavioral profile of these individuals having reached adult age should allow us to provide stronger conclusions. Finally, I would like to thank my supervisor and our technical team for their great help in the conduct of this project. And thank you for viewing my virtual poster presentation. Please do not hesitate to ask me any question in the comment section.